Cancer Genomics and the Future of Public Health, April 2019, by Kathy Marchant, Louise Montgomery, and Blessing Ugamba. This is a synopsis in 3 parts. The first part is the evolution of cancer genomics and the data. The second part is sequencing populations to precision medicine. And the third part is current applications in cancer genomics. Cancer is the second highest cause of death in the United States, with 23.1% of all deaths. It is second to heart disease. More men have died from cancer-related disease than women in the United States. African American men have the highest cancer mortality rate in the U.S., followed by white men, African American women, and white women. The Human Genome Project sequenced about 30,000 human genes within our 23 chromosomes. Now we have organizations such as the Sanger Institute creating the Cancer Gene Census of Somatic Mutations in Cancer, or COSMIC, a database of genes strongly implicated in cancer. This model of the BRCA1 gene was generated by COSMIC 3D. The BRCA1 gene is only one of the genes associated with breast cancer. Originally, it was hoped that a single genetic mutation could be treated to cure cancer. Unfortunately, genomes of cancer cells have shown many genetic mutations that can lead to the same cancer in different patients. ENCODE is the Encyclopedia of DNA Elements and is a project from the Broad Institute of MIT, UC Santa Cruz, and Stanford, and it catalogs the regulation of DNA by other elements within DNA. TP53 is a gene, and this is a rendering from COSMIC 3D. One common process of regulation in cancer genomics is called methylation. An example of methylation is the activation of p53, a protein active in tumor suppression, which is responsible for turning on or turning off tumor growth. The Human Genome Project sequenced genes. COSMIC catalogs genes implicated in cancer. ENCODE catalogs the regulation of DNA. Within DNA, researchers identified over 3 billion base pairs sequences, or nucleotides. Common sequence changes called single nucleotide polymorphisms, or SNPs, are being analyzed between populations for mapping the location of nucleotides called alterations causing disease. HapMap is one project identifying differences among populations using samples from Nigeria, Japan, the United States, and China. Researchers commonly compare SNPs among patients with a particular cancer to those without the cancer, seeking to map the location of cancer within the genome. These types of projects create data found in repositories all over the world. This is a database SNP from NCBI. NCBI hosts probably close to 100 different databases about the genome and genes. And finally, these are 2 more areas of research. Genes are altered by many environmental factors, and researchers are tracking these biologic pathways causing DNA changes. And finally, pharmacogenomics is the study of how genes affect a person's response to drugs. These are some resources for cancer genomics. There are hundreds of repositories for data. These resources are for well-known cancer research institutes around the world. Cancer Moonshot is a funding opportunity from the National Institute of Health, requesting proposals to create a new infrastructure called the National Cancer Data Ecosystem. The hope is to increase collaboration and make available emerging therapies to cancer patients, reducing impact globally.
The study of cancer genomes has greatly improved our understanding of the biology of cancer. So, will genomics cure cancer? Well, the most rapidly advanced field in life sciences has been genomics. And a famous biogeneticist, Dr. Eric Lander, quoted, "For the first time in history, we can finally pop the hood and see what's going on. We are entering a period where we are able to get a complete catalog of disease genes." It took almost 15 years and $3 billion to get one sequence, the Human Genome Project. Over the last 12 years, we have looked at thousands. Although this is not a cure, but well on its way. The wealth of data emerging from studying cancer genomics is being integrated with patients' medical histories and clinical data. These integrated results are then being used to develop more tailored approaches to cancer diagnosis and treatment, as well as to improve methods of predicting cancer risk, prognosis, and response to treatment. This has led to precision medicine. According to NIH, precision medicine is an approach to patient care that allows doctors to select treatments that are more likely to help patients based on a genetic understanding of their disease. This is also called personalized medicine. In 2015, President Obama unveiled the Precision Medicine Initiative. The mission of this initiative is to enable a new era of medicine through research, technology, and policies that empower patients, researchers, and providers to work together toward development of individualized treatments. The Precision Medicine Initiative is a long-term research endeavor that has both short-term and long-term goals. The Precision Medicine Initiative is a $215 million project that is based on the premise that taking into account personalized information about genetics, environment, and lifestyle while making treatment decisions will improve the precision and success of patient care. The initiative has 5 main objectives. Number 1, more and better treatments for cancer. 2, creation of a voluntary national research cohort. 3, regulatory modernization. And 4, public-private partnerships. The idea of precision medicine is not new. Healthcare providers have always focused on delivering new and effective ways to treat their patients. However, with the tools and techniques of 21st century genomics is starting to replace older methodologies. This precision medicine cohort is also known as the All of Us Project. This requires research, clinical trials, and multidisciplinary collaboration. The PMI cohort is the cornerstone project of this initiative, and the goal is to gather genomic and background data from at least 1 million patients to give big data researchers something to build on. This initiative will change the way we do research. Participants will be partners in research, not subjects, and will have access to a wide range of study results. The PMI cohort won't just gather an unprecedented amount of data in one place to be shared across the research community. It will allow researchers to explore a more accurate portrait of patients who experience a variety of comorbidities, conditions, socioeconomic challenges, and lifestyles, something not always accessible from strictly controlled clinical trials. Basically, we're changing the way we do research. So a few of the benefits associated with this initiative are improved diagnosis and treatment, targeted therapies, and screening. Currently, every baby born in the United States is tested at birth for between 29 and 50 severe, inherited, treatable genetic diseases through a public health program called newborn screening. This whole genome sequencing would enable clinicians to look for mutations across the entire genome simultaneously for a much larger number of diseases or conditions. Some of the challenges and risks associated with this initiative are data security and privacy. Because of the remarkable advancements in genomics research in recent years, the use of genomic testing has gone from rare to routine in many clinical settings. Yet, while there are clear clinical benefits to the patient, this introduces new risks to patient privacy, and patients are potentially vulnerable to the misuse of their genetic information. To address this, in 2008, Congress passed the Genetic Information Non-Discrimination Act to restrict the access of issuers of health insurance and employers to individuals' genetic information, as well as prohibit genetic discrimination. Additional challenges of the initiative include cost, as well as misinterpretation of genetic and health data. A collaborative clinical genome research program called ClinGen has been initiated to promote transparency and advancement in genomic research. Projects such as ClinGen address and seek to reduce the risk of precision medicine over time. Also, there are ethical concerns, such as safety, embryos, and genome editing. So, for example, is it okay to use gene therapy on an embryo when it is impossible to get permission from the embryo? Is getting permission from the parents enough? What if gene therapies are too expensive and only wealthy people can access and afford them? That could actually worsen existing health inequalities. Will some people use genome editing for traits not important for health, such as athletic ability or height? Is that okay? These are things to consider. The National Human Genome Research Institute is responsible for considering the ethical, legal, and social aspects of genomics research in our work.
Genomic tests examine the activity and sequence of genes within a sample of tumor tissue to provide unique information about tumor biology that other clinical pathological factors simply cannot. The activity and interaction of genes influences the behavior of a tumor, including its aggressiveness and how likely it is to grow and spread. Lung cancer. Lung cancer is the most common cause of cancer death worldwide, with an estimated 1.6 million deaths each year. In both men and women, lung and bronchial cancer accounts for the highest estimated cancer death rates in the United States. It is also the second most recorded cancer incidence case in both sexes. Each year, more people die of lung cancer than of colon, breast, and prostate cancers combined. NCRI and collaborators have developed the proposed delivery of synthetic lethal approach. Basically, a genetic-based approach and a drug-based approach are used. This is termed targeted therapy. Lung cancer screening. Early detection can lower the risk of dying from this disease. Sputum cytology, chest X-ray are routinely done to screen for lung cancer. However, the recommended screening test is the low-dose CT scan. In this test, an X-ray machine scans the body and uses low doses of radiation to make a detailed picture of the lungs. LDCT is mostly for people who have a history of heavy smoking or smoke now or have quit within the past 15 years or are between the age of 55 and 80 years old. Many studies profiling the genomics of lung cancer have discovered that the most frequently mutated genes were the EGFR, the TP53, the ALK, and the KRAS. Comprehensive genomic profiling of lung cancer is rapidly becoming an important trend in cancer pathology diagnosis. Breast cancer is the most commonly diagnosed cancer and the second leading cause of cancer death among women. The incidence and mortality rates also differ by countries. The US has the highest incidence rate and China has the lowest incidence rate. Denmark and Netherlands have the highest mortality rate, while China also has the lowest mortality rate. The breast is made up of three main parts: the lobes, the ducts, and the connective tissues. The lobes are the glands that produce the milk, the ducts are the tubes that carry milk to the nipple, and the connective tissue surrounds and holds everything together. There are several kinds of breast cancer, but the two most common types are the invasive ductal carcinoma and the invasive lobular carcinoma. And just like their names, the ductal carcinoma are when cancer cells grow outside the duct, while the lobular carcinoma are when cancer cells spread from the lobus to the breast tissues. I think we all have heard of breast self exam or clinical breast exam in one of the breast cancer awareness programs. The US National Cancer Institute recommends that women initiate a biannual screening of mammography at age 40 to 49, and this should continue annually after the age of 50. Now, research has shown that high penetrance genetic mutations in BRCA genes, the P53, the P10, the STK, the CDHI, and a much larger number of moderate penetrance variants in the CHK2, the ATM, the RAD51, and the BRIP1 predisposes one to this disease. More recent studies have also identified PALB2 genes, that's the partner and localizer of BRCA2 gene, as a bona fide breast cancer susceptibility gene, and recommends a genetic testing in patients with hereditary breast cancer along with BRCA status. Post-biopsy, the Oncotype DX is a test given on tissue samples which analyzes 21 genes in patients with an estrogen receptor positive breast tumor. This test predicts response to chemotherapy as well as chances of cancer return or recurrence. The Oncotype DX tests are translated into a recurrence score, providing a personalized information which enables doctors to predict which women should undergo or will benefit most from chemotherapy. Prostate cancers are cancer cells originating from the prostate. The male reproductive system includes the penis, the prostate, and testicles. The prostate produces fluids that make up a part of the semen. An increased prostate causes the urethra to narrow and decreases urine flow. Prostate cancer is more common in African American men than in white men. It is the most common cancer case in men and the second leading cause of death by cancer in men in the US. The most common risk factor is age and family history. Prostate cancer screening. Some of the tests that can be used to screen for prostate cancer include PSA test, a digital rectal exam, a transrectal ultrasound, and post-biopsy a Gleason score and an Oncotype DX genomic prostate score. A mutant gene that alters the androgen receptor was discovered in African American men with a family history of prostate cancer. Recent molecular studies show that prostate cancer tissues overexpress both the 5-alpha reductase 1 and 5-alpha reductase 2 isozymes, whereas benign prostatic hyperplasia tissues overexpress only the 5-alpha reductase 2 isozyme. Biopsy alone often misses patients with high risk of adverse pathology. For example, tumors in approximately 60% of patients with a lung adenocarcinoma have been found to be linked to specific genetic mutations. Personalized medicine involves looking at the cells obtained from a biopsy to see if there are any genetic mutations that could be linked to the type of cancer a patient has. This is because certain chemotherapy drugs are either more or less effective than others against tumors with specific mutations. Molecular analysis of a tumor, which is also called genomic testing, can help determine which therapies will be most likely to benefit a patient. Research is currently ongoing on cancer genomics and precision health medicine. Thank you.